Last time we were talking about um, these steps in pathogenesis. So typically what you have is you have exposure to the pathogen. Obviously, you have to be in contact with it. And so whether it's a cell or a virus, it's going to have to not only be exposed, but then it has to bind. And so then you have a binding event. So this is going to, what's going to be required here is there's going to be specific interactions between the cell membrane of the protein, of this microbes or the viron, right? In both cases, you have a protein-protein interaction at the cell membrane interface. And then you have invasion. So if, depending on the type of microbe, there can be a lot of multiplication, lots of colonization. The book interchanges the terms multiplication, colonization, and same concept. So invasiveness would mean further growth. And so whereas all microbes and viruses will multiply inside, the difference here is what is causing the disease process. So you can have a microbe that doesn't elicit a lot of multiplication inside. It's just kind of only stays localized. It doesn't grow and damage other tissue, but it will release toxins that will damage surrounding tissues. That's different, right? That's different than a cell growth mediated event. Instead, it's going to be a toxin mediated event. So, or you can have invasiveness where you just have growth and just your cells are their food, and so you're dying at their expense. And so this is a bad pathogenic. In both cases, tissue or systematic damage is observed. I think we understand that. And of course, we have here, this is just a review. We have first was exposure. We have adherence. Adherence is we have the cell membrane of the host cell is going to have a receptor that bacteria cell, in this case, is going to bind to. Once you have this binding event, then the bacteria can work on reversing that cell membrane. The hardest part is to get the binding event to occur. So we have many structures that can function in this capacity. Here, what we have is that protein is binding to a cell receptor. This is a simplified diagram because we see a cell and then we see an adhesion molecule coming from the bacteria. But where is this adhesion molecule anchored into? What is it bound to? So you can have it bound to just the cell or the viron, as we've observed, but you can also have it as a part of a capsule. So a capsule is, and I normally would write this on the board and we would get into this in a little more, but a capsule is a bacterial extracellular polysaccharide. I'm going to say it the way my notes have it, how the book has it, just this is my definition. Bacterial extracellular polysaccharide structure that covers the outer layer of the cell that can bind, okay? So it's like a covering. And here you can see that we have in dark blue or purple, I don't know, I can barely go beyond the primary colors, but here we have a cell, and here you have this more light purple region. This is the capsule. This is a polysaccharide extracellular matrix. This serves as a barrier for the host's defenses, against the host defenses. So the capsule is a bacteria extracellular polysaccharide structure that covers the outer layer of the cell that can bind host surface receptors and protect bacteria from host defenses. So this is almost like a unicellular slime layer, if you will, or something like that. You can look at it like that, but every single cell has its own capsule. Okay. There's other structures that can, here we have more capsules. Okay. So we can see that this is obviously protecting the cell. So if your immune system's trying to get to this bacteria, it has to negotiate with all these extracellular polysaccharide structures. So we have other structures. We have fimbriae, flagella, and pili, and they can also function as attachment. Here we have, I think this is computer generated. I don't think this is what they see, but it looks really dramatic, right? It looks like I'm looking at an episode of Battlestar Galactica or something. Anyway, so here we have pili, all right, and flagella. I'm sorry, fimbriae. We don't have the pili, that's for conjugation. We have fimbriae, which are tiny hair-like protein extensions, and we also have flagella. So these things can bind too. These can have receptors. And so you can imagine that species have variations where they do or do not have this fimbriae, do or do not have this flagella, which means they do or do not have the structures necessary to be very virulent, okay? Virulent. My wife keeps telling me to correct myself. All right, so that could be a difference. So you have a species, and depending on whether or not it has these structures, really can dictate whether or not it is virulent. Okay. So then we have colonization. Colonization is microbe establishes in or on a tissue, and then it multiplies. And it can be harmless, like skin flora. It doesn't need to be thought of in terms of pathogenesis. It could just be, you're exposed, you adhere, and then you colonize. So we're not at pathogenesis yet. When you get to disease is when you got pathogenic issues. And so colonization is just establishment of the microbe on a tissue, and then it multiplies. It can be harmless like skin flora, but it can be pathogenic um, depending on the portal of entry. So if you have Staphylococcus and it's sitting on your skin and it's just a part of your normal flora, great. But then you have a puncture wound, a stabbing event, and then now what was on the skin has access to the deeper tissues. This can become pathogenic. So if you change the portal of entry, you can change whether or not it is benign or pathogenic. And normal flora is a really big barrier to a lot of pathogenesis. Normal flora are favored and provide a barrier from unfavorable microbial residents. Obviously, we need to have bacteria on our skin, and when bacteria that we don't want land on our skin surface, they really are landing on a coat of bacteria. These bacteria don't want to be hanging out with them, and so they are providing defenses for us. If you have so that's the skin, but we can scan our body and then kind of realize what portal of entries occur anytime that there's a difference from where the skin is, right? So if you start from the top of your head and you start to scan down, you have your eyeballs, that's a portal of entry. You got your nostrils are portal entry, that's the upper respiratory tract. Your mouth is the same thing. Your ears, okay? And then as you go down, you have the urogenital area, and then you have anus. And so these are the major portal of entries. So every one of these portal of entries needs to be structured so that it defends against unintended microbial growth. In the upper respiratory tract, we have mucus. Mucus is naturally inhibits microbial attachment because it's a fluid, it's moving, it's constant. So it's hard for the microbe to get access to the epithelial cells if the mucus is there. The mucus is just a fluid of polysaccharides, and some microbes can establish residence even if they can adhere to epithelial cells anyway. So some microbes can break through this mucus and adhere. And so here we have in figure 25.6 is microbial cells are landing on the mucus, but then they can penetrate through the nucleus, through the mucus, not nucleus, and then they can start to multiply. This can lead to pathogenesis. Now we're going to talk about, give me a second here.
We can't hear anything.
Okay. Just give me a second. I have to open up a new file. My computer had shut down, did not save my previous PowerPoint. I know it's supposed to be auto save. So I have to mobilize a slide here. Okay. Thank you for your patience. So frustrating because I worked so hard on all of this and then it was just erased. But anyway, here we are. So you can see this tooth. So we're going to talk about dental caries. I'm going to pan off. I'm going to pan back to my whiteboard in a minute. I just wanted to kind of cover the basic structures of tooth anatomy. Okay. We don't need to know too much about this. The big thing to pay attention to is where we have a portal of entry. And so, yes, you have your mouth as a portal of entry into your intestinal tracts, but your teeth, because of the tooth structure, are also a portal of entry. And so you can see here, we have a lot of live living tissue. We can talk about it in terms of tissue, bone marrow, periodontal membrane, alveolar. We don't need to know all those structure. But what's important here is the gingiva, and then here is really what I want you to focus on: this gingival crevice. I don't know if I'm saying that right. I know
Gingiva is how is that, but then yeah, gingival is what I've heard.
Gingival depends if you're from England. So gingival crevice is right here, and so this is an interface between the enamel, which is not living, and blood supplied tissue. And so we have this crevice here that can be exploited. This is a three-dimensional concept, not a two-dimensional concept. So I want you to just remember that at all times. And so what you want to think about is what is it going to take to get into this living tissue from around here. It's pretty protected, actually. And so there's a calcification matrix that makes up the enamel. If we have a break in the enamel that goes into the dentin, then you're going to have access to nerves and stuff like that. You're going to get pain. And so what compromises this structure of the enamel? Let's talk about that. Hey guys. All right. So I actually, unrelated, I was talking to an oral surgeon friend of mine. We were just lamenting on this current situation and what it means. And then I used the opportunity to talk to him about some of the anatomy of the tooth, and this is how he explained it to me. He knows it in a way that hopefully sounds friendlier than me. So your mouth is full of lysozymes. Lysozymes are have antimicrobial activity. You also have that in your tears, and so this is defenses. And so when you're, your mom, when you have a cut, and your mom would, kind of lick her thumb and put it on there. Somehow that's been evolutionarily selected for. I don't think she understands the lysozyme presence in your saliva, but this is actually kind of helpful. Or if your dog licking a scratch on your leg. So that the saliva has antimicrobial activity, but it also has a lot of polysaccharides and proteins, and so this can serve as a nutrient source. So your saliva enables your teeth to be a nutrient source, because unlike your mouth and your nose, your teeth aren't really covered kind of the same with mucus. So there is always going to be bacteria on your teeth. It's a constant presence of bacteria. The point of flossing and brushing deal with trying to minimize the amount of growth that has occurred. You can look at it as kind of controlling a biofilm, because that's really what it is. So on your teeth, you have a biofilm, and you guys see this, okay? Okay. So we're going to be talking about dental caries, and this is another way of saying tooth decay. All right. So saliva on the tooth surface provides microbial attachment site. So saliva, because of the nutrients on the tooth surface, makes a great attachment site. So this can be harmless, or you know, and if it's kept in check. So we'll say attachment site for Streptococcus. Okay. And Streptococcus, we have two main species. Hold on, let me organize this so that I can. We have two subspecies. We have Streptococcus sabrinus and we have Streptococcus mutans, and they both contribute in different ways to be able to establish a biofilm on the tooth surface. And so the sabrinus contains capsules with adhesions. Adhesions, as you remember, have binding capacity. Now, I wouldn't say that the tooth surface has the cell receptors in quite the same way, but we can still think of the capsules as having proteins that are able to engage in the enamel structure and hold on. These both are in the same biofilm, so whatever sabrinus is bound to, mutants is bound to, and mutants produces dextran. That I guess we can just say penetrates crevices and fissures. So this is going to kind of go into all of those areas. Your tooth surface is not smooth. I don't want you to think of these crevices as injuries. It's a very rough topography on your tooth surface, and so there's a lot of opportunities for mutants to put dextran and exploit this. And so once they're bound there, now they're just sitting, like what we were talking about with biofilms, they're mobilized on a surface where nutrients are constantly passing over. Kept at this rate, everything is okay. This is actually not bad. Because I don't have a lot of room, I have to erase. I would normally like to keep this as a continuous concept. So Streptococci, you have to remember this is a biofilm. The biofilm is going to encapsulate the bacteria, protects it, and inside of this biofilm, oxygen is sequestered right away. And so it doesn't take long for the biofilm to becoming an anaerobic environment. It's an anaerobic microenvironment. This is really the key to how you get gum disease. Because if you continue to harbor and cultivate an anaerobic biofilm environment, you're going to continue to have biofilm growth, and then you're going to get more of fermentation. So Streptococci ferment glucose to lactic acid. And then lactate. Lactic acid, if it doesn't have the proton, it's called lactate. Lactate destroys the tooth enamel. Okay, so this is really where you get in trouble. So depending on how far this goes, this doesn't happen right away, but if you have sweets right before bed, or if you have a glass of milk right before bed, and you allow that sugar to be on your teeth, and then you are sleeping, you're not eating a lot of food, you're not drinking water or anything, this becomes an immobilized environment. And then you're breathing through your nose all night. And so now there's a real good opportunity for this biofilm to continue to exploit all of the food and then erode at your teeth. That's why you want to brush before bed, because you can remove some of this biofilm, and you remove all the nutrients that came from your food and whatever you were drinking, and then it slows down biofilm growth and promotes the tooth health, not promoting tooth decay. All right, and so if you could, but if you don't do that, then streptococcal growth continues. Now your biofilm is getting more and creates dental plaque. So this is where we start to get in trouble. We're not in big trouble yet. We wanted to keep it at the biofilm level. Continue to grow, you'll get to a certain density, you'll have a dental plaque. And this is a different level of biofilm. And as we remember, biofilms contain a community. Communities are exploiting a niche. And so communities have many different species. And so once you allow for this dental plaque to come, you promote the growth of a lot of other bacteria to come in there. And this is where we get in trouble, which promotes the growth of, okay, who's coming to the party? We got a nice dental plaque coming. Well, we have Corneobacterium. Bacterium. It's probably one of the species I know least about. I should say genus. Leptotrichia. This is just some of them. I know there's more than this. The book doesn't list them all. Neisseria, Fusobacterium. This is just to name a few. I know of at least 3 other species, but we won't have to get into all of them. And so we start off with Streptococcus mutans and Streptococcus sabrinus. We establish a very small little biofilm. They are on there growing. If left unchallenged, this biofilm becomes a dental plaque. This dental plaque will then promote and protect the growth of all these other species. So now we have a big party going on your teeth. And with more species, there's more microbial activity, there's more fermentation. And so the biofilm produces lactic acid or lactate that decalcifies the tooth enamel. Now we have damage. Okay. And producing dental caries. My arm is extended, so I get more sloppy. Okay. So we started off with just a normal, very subtle biofilm. With the Streptococcus, if Streptococcus growth continues, you should form a biofilm called a dental plaque. That's stage 2. And then if that dental plaque goes unchallenged and remains in place, then it continues to grow. This biofilm then promotes and supports the growth of all these other bacterial species. Now you have numerous bacteria inside of this biofilm. This intensifies lactate production, and this will decalcify the tooth, and then you have a dental carie. And so, and I won't write this down. And so what you're doing when you're flossing, you brush and you kind of taking off the nutrients and all these things. But you're more taking away a lot of the substrate for them to grow, but you're not taking care of the biofilm. Biofilm, and then when you do deep cleaning, I guess they do take care of some of that. When they scrape and all that stuff, they're trying to take that biofilm off. Flossing, what it does is, let me share my screen. And so what flossing does, flossing gets in between this. Remember, this is a 3-dimensional concept. This is a 2-dimensional image, but this is all around. Flossing gets in there, and what it does is it introduces oxygen into the biofilm. And so the biofilm requires an anaerobic condition. So when you disrupt not only the structure of the biofilm, but you introduce oxygen, you create an anaerobic environment, and that disrupts all fermentation, and that stops the production of lactic acids. That's why they're always telling you to floss, because it's not just about brushing. If you get brushing, that's fine, but what about this area right here? And any of you want to date a dental hygienist, and you have really nice teeth, well, some of these ladies I know that are dental hygienists, they're going to be looking at your gums, guys. They really know that. That's the key. And so the gums is a really good indicator of how the health is at this interface. And you should take a look in the mirror, and you shouldn't just be looking at your white teeth. You should be looking at your gums, because your gums are going to I mean, look, the tooth is anchored in the gum. If this tissue is not okay, this whole tooth is not okay. And you can have healthy teeth by taking care of your gums, and you can take care of your gums by flossing. Okay, that was my dental tip for the day.
Can I add that unwaxed dental floss is better than waxed dental floss?
Kidding? Oh, because it produces because it lays down a layer for biofilm growth, I guess. Never thought of that. Yeah, that's true. You're helping the biofilm. Here, have some wax. Here's some substrate. Very nice. I never thought about that. That has to be true.
Well, there are a lot of studies to support that. I'm sure there are several reasons, but like I said, wax as well. But also there's more friction with unwaxed as well. Being able to get more maybe material that could promote growth, food as well.
Well, it seems common sense that if you put a layer of wax on top of biofilm, that's bad news, man. All right.
I never thought of that. It's great.
Yeah. So, I mean, there's some techniques in culturing bacteria where wax is used in that benefit. So I just don't want to do that. Okay, so let's go on. So invasion and systematic infection. So invasion. And so all of this stuff, guys, you need to know. I know I've been writing on the board a lot. I don't have time to go through a lot of this. The PowerPoints do have a lot of these concepts that I really want you to know. So here, let me boom, boom, boom. Wow, now it looks all dramatic. Very nice. Common concepts here. So invasiveness, ability of pathogen to grow in host tissues at densities that inhibit host function. So that's a distinction between going into the tissue, being at a density that doesn't disrupt host function, and one that does. I'll talk more later about the difference. So that's not necessarily invasiveness just because you've gotten in there. You have to grow to a density that really makes things a problem. Bacteremia is the presence of bacteria in the bloodstream. So anytime you have a portal of entry that's through puncture, you're going to have this, but it also can happen in your upper respiratory tract any of your portals of entry can be this way. And this applies to viruses too. A lot of times when you're dealing with sexually transmitted diseases, a lot of times the portal of entry is thought of to be just the genital surface area, but that's not necessarily true. With the friction that comes with intercourse, you create these little micro cuts, and then this is a portal of entry to the bloodstream. This is really important. But that's not the same thing as septicemia. This is when there's an actual exploitation of the blood. Remember, blood is tissue. So blood-borne systematic infection may lead to massive inflammation, septic shock, death, all the good stuff. So be able to understand the distinctions there. And I don't like that they put this definition here, but it is here and it's in the book, and so I kept it. Infection, as we talked about before, is just establishment of microbe. It is not necessarily pathogenic, but it is lumped in with the concept of colonization. So remember that if infection doesn't lead to invasiveness, then you don't have pathogenesis. All right, let's talk about, I'm saying bye to my wife. We'll talk about virulence. Pathogens use various strategies to establish it. Hold on one second. You okay? All right. And we want to determine the extent from which a pathogen is effective, and so we call this virulence. So not pathogenesis, or how pathogenic is this bacteria? How virulent is it? It's not considered pathogenic. That's just not the right way. Pathogenic means that you're talking about the host being hurt. That's just not the same thing as measuring the capacity of a pathogen to impart disease. So this distinction is really important. So we measure virulence by lethal dose 50. The LD50 is the amount it takes to kill 50% of an animal in a test group or a population, any kind of population. So highly virulent pathogens show little difference in number of cells required to kill 100%. So here we have a comparison of two different bacteria. And so we would say, how many cells does it take to kill 50%, right? And so if we were to measure the LD50 of Streptococcus pneumoniae, we would use this number, which is, between 100 and 1,000. Not sure what exact number, but this would be the LD50. It takes more cells of Salmonella to inflict the same damage, to kill 50% of the population. So if you have a lower LD50, you are more virulent, okay? Virulent, I think. I don't know which way the right way to say that medically is. But that's the distinction. And so if you see on your exam pathogenesis of a virus, that's not the right way to say it, right? How virulent something is. And so in this graph, you have to be able to interpret this. We would know that Salmonella is less virulent than Streptococcus, okay? And so when you're measuring virulence, you want to think about what it is about this cell that makes it virulent, okay? And so you can have subtypes in a species that make it virulent. And so here, if you remember from chapter 1, we had an example of transformation. We had Streptococcus pneumoniae, and we had an R strain and an S strain. This R strain does not kill mice, S strain does. And so, this is actually worded wrong here. The rough stain does. And then what we did is you can see, so you could mistaken this as two different species. We here have, Streptococcus pneumoniae different strains. Oh wait, you can't see my laser pointer. So here we have Streptococcus pneumoniae, we have two different strains. You could say this is two different species, but if we kill the S strain and then we incubate it with the R strain, which previously couldn't kill the mice, and then we introduce that transform R strain, then it kills the mice. So what this says is there's some genes within the species that can impart virulence. And so this ability to kill is just due to some difference in genes, but these genes are not essential for bacterial growth. Okay. Any questions there? So what we want in order to make a vaccine is we want to have an attenuated version of whatever we're looking at. And so you could take, so we have the S strain that killed. And so, well, how could we make a vaccine? This microbe is going to propagate through killing the mice and then using the mouse as a substrate and then continuing to spread like that. Pandemic, bubonic plague is, bacterial based. So in a way, what you have is the survival of this particular microbe being dependent upon whether or not it can cause disease. So those genes, those pathogenic genes, are going to remain. What you do when you want to make a vaccine is you take this S strain and then you just put it on normal media and you continue to plate it, and you're taking away the selection for those pathogenic genes, but you're maintaining all the other things that are on the cell surface of the bacteria. And this becomes an attenuated version of that strain. And so it doesn't actually cause disease anymore. So then you can introduce this attenuated strain into an organism, and then this organism will elicit an immune response against that strain. And then when the virulent strains come in, you already have antibodies to it. This is the basis for vaccinations. You can kind of make proteins, you can make the S spike protein for the SARS, virus, and then hope that works. But a better way would be just to take the existing COVID virus, the SARS virus, and then just continue to cultivate it without allowing it to propagate through the disease process. And then what you do is now you're deselecting against its pathogenic properties, virulent properties, and then maybe you get a version of the virus that no longer elicits disease. This is part of why they can't really tell you the extent. I'll stop sharing because I'm talking. This is part of why they can't tell you. There's so many unknowns about this COVID outbreak, right? One of the things they don't understand is there's no symptoms for some people that they can spread. We don't know that, right? Maybe some people can get it and then they don't spread because there's a lot of coronaviruses of the common cold. Maybe you've had a presentation of coronavirus that really mimics a lot of the structures of the SARS-CoV-2, but we don't know which one of those cold strains it is. So maybe it depends on that. There's some cross-reactivity though in species. And so the whole goal is to try to create a unique vaccine that takes care of this particular COVID virus. And there's a lot of the anti-vaxxers that will be like, oh, but you're introducing disease and all this stuff. I don't think I have to defend the utility of virus therapy and vaccines, but obviously this is much less, even if it does elicit some immune response. I mean, that's the whole point, right? We want to elicit an immune response. We just don't want you to succumb to disease. So if you get a little bit of I feel a little bit off for a couple hours, that's okay. It can happen sometimes, but you're not getting the disease, and that's the point. You're creating an antibody response for the real thing. All right, so the virulence of these different species can be thought of as existing on different regions. And so these will be known in some cases as pathogenicity islands, all right? And so check this out. This is a cell of Salmonella, and these are all the different proteins that enable it to inflict disease on its host. You have enterotoxins, cytophores. I don't want to get into all these, but just know that every one of these proteins has a gene in it. These genes are usually clustered in the same region, and so we have a name for this. We call them pathogenicity islands. You look at the whole chromosome. Let me. If you were to look at the whole genome of Salmonella, the Salmonella genome, 5 prime, 3 prime, and then you have all your genes. Maybe here in this region, and then maybe here in these regions. If you took these regions out, maybe there would still be, fine cell growth, no problems. But these regions, we notice that they have all of the proteins that inflict disease, and so these are known as pathogenicity islands. These are just regions. This is one of the reasons why scientists do not think that COVID-2 was a bioweapon, because, as I said before, it's poorly designed. They could do much better. If you were to really be evil, you would have engineered this COVID-2 and you would have just randomly placed something like Salmonella pathogenicity islands in there. Really unload. There's all kinds of, you could harvest pathogenicity islands from a number of species, pack them into one genome, and then deliver that with a virus. That would be horrible. So as it turns out, COVID-2 has very subtle mutations that lead to a big difference in its morphology. Okay, well, at least it's morphology. It binds to completely different receptors, but it's only 1 or 2 nucleotide difference. That's why we don't think that that's possible. Okay, it's just consistent with an evolutionary trajectory of a viral genome. Okay, where are we going next? Okay, so we have attenuation. Obviously, the host health is also of concern, and this is what still remains to be seen with the COVID-2. So there's a example of a virus that rabbits caught in Australia. The book has some of this, but I remember reading this when I took pathogenesis and microbial pathogens in graduate school. This virus that killed all the like 90% of the rabbits in Australia. Now, if you were to measure after 2 weeks, it wasn't 90%. It was something like 2%. But if you measured after a year, 90% of the rabbits were dead. So the compromised host is something that other opportunistic pathogens can exploit. And so there's documented 25% respiratory capacity for people that recover from COVID. So we'll have to see how well they do for the rest of the year. You've just become heavy smoker category. We know that depriving all the vital organs with needed oxygen has a detrimental effect on your health. So a compromised host can occur in all of these cases. And so that's a big part of the pathogen-host interaction. So I worry about people that are recovering from COVID and whether or not they're more susceptible to other diseases, pathogenic or otherwise. Okay. I don't want to really talk about this too much. This is just a concept of enzyme destroying enzymes that destroy host tissues. They specifically will destroy different host defenses. And so here we have an example of Streptococcus pyogenes. And this has an enzyme that will penetrate the extracellular matrix of epithelial cells. And so then they have access in a way that other cells would not. So because of this extracellular enzyme, they're able to penetrate all the way through into deeper tissues, continues to eat away. This is a cool one. Look at Staph aureus. So here we have blood cells And what Staphylococcus does is it produces coagulase and basically induces clotting. And when you have this clotting, you now have protection against the cell being able to, it forms kind of a wall from the immune response of the host to be able to access these cells. And so then Streptococcus can then go in there and continue to exploit the tissue. So, and the only thing is that this diagram doesn't do a good job. I wish they had one step before the platelets. So if you can imagine this blood vessel and that these little structures are gone, okay, on the side, and it's open there. So if you were to have Staphylococcus enter the bloodstream, your immune response would see it, clear them out. But what Staphylococcus does is it really induces a fake clot, and then these clots form on as just a little. It doesn't, obviously, if it blocked off your entire vessel, that would be a bad event. But enough partial blocking where there can be a protection against an immune response and a little bit of a chance to divide in your numbers. And so here you have unblocked thing. Oh wait, I think I'm reading this diagram wrong. Maybe these are the white cells. I don't know. They don't really diagram this much. But here you have the cells that produce the coagulase. Then you have the clot and these immune responses. Yeah, I think that's the way it was. I'm sorry. I don't look at this diagram a lot, and I don't usually cover this concept too much. So I'm a little confused on what the details were. So I guess the clotting is happening here at the Staphylococcus culture or population, if you will. And then it forms a wall around here, this localized. And so now I guess these are immune cells. Yeah, it's labeled here. Immune cells can't access these streptococcal cells because they're blocked off with this wall. And then we get the cells are able to be released into the bloodstream. I was a little confused about the different colors that were shown here as purple. They should have just kept it the same. I guess they're trying to flex that they're gram-positives. But anyway, that's very minor. What I really want you to think about is this AB type exotoxins. Okay. These are really cool. AB type exotoxins. Two subunits. Hey,
Dr. Cruz?
Yes.
Your camera is not. Oh, yeah, there you
How about that? See that okay? Okay, so we have two subunits,
Yes.
A and B. So B binds to the cell surface. And A enters the cell and causes damage. An easy way to kind of remember this is you have A and B, right? So you can say the A subunit attacks and the B binds. That's an easy way to kind of remember this. So this is a class of exotoxins. And so one thing about these exotoxins, this is a different way to inflict disease. This is not a mechanism of pathogenesis that results from proliferation of the cell within the tissue. Usually these AB exotoxins based bacteria don't leave the original site of wherever their portal of entry was. So if you have a puncture wound, tetanus, Clostridium tetani is a bacteria that can like just enter. But if you were to succumb to the disease of tetanus, It's not because this bacteria grew systematically. It's because the bacteria produced a toxin that led to systematic disease. Okay? So that's different than what we were talking about before with septicemia and all that stuff. Those diseases are from the actual bacteria using your body as a food source, and they're growing at such numbers that it's hard to tell whose body this is anymore. This is different. These exotoxins, it's almost like you can think of these things like hormones. They produce a molecule that has an effect at a distal site, and then that disrupts physiology and health of the cell. Okay. The toxicity. This is a different concept. So this is ability of a toxin to cause disease that inhibits cell function or kill host cells.
Can I clarify just curious? So to define the two, the AB exotoxins basically produce a secondary a toxin of some sort as a byproduct, and so that's what causes the damage to the host on the host cells. And contrary to that, septicemia is when there are such a mass production of microbes and bacteria that the nutrition is being stolen and the cells are being damaged by the actual bacteria itself.
Yeah, and a lot of times septicemia will, okay, I guess I wasn't clear on that. It's not just the cells though. It's the immune response. It seems like the immune system, it can seem a lot of times that the immune system betrays us. You know, and our immune response is really what kills us in a lot of these COVID cases and stuff. Your immune response is happening all the time. It's totally on your side and it's totally working. You have no idea. Newborn babies are born in hospitals. These are places of disease. It's been challenged hundreds of times before it left the hospital door. But its immune system is cranking and neutralizing all kinds of things. So our immune response is always, it's just working beautifully at all times. When you have sepsis and you get bacteria in the blood, your immune response, you just start to freak out. It goes into overdrive, and it's just not to scale that you can handle. And so causes damage through response, but not through a specific molecule. You understand the difference? Like it's not like the bacteria is producing a molecule to elicit that response. Its presence is being detected by the immune system, and then the immune system is into overdrive, and then you go into septic shock and you can die.
I see what you're saying now. That's the difference. It's the immune response causing inflammation and possible leakage into tissues, et cetera, et cetera.
And this can vary, right? I mean, if you get cholera, which is an AB exotoxin, like you're not going to vary your response to that, right? You get cholera, you got cholera, man. You're gonna be going to the bathroom a lot, and so that's not host specific, right? But people have different reactions to microbial invasion. Some viruses wipe out, I think I might have told you this before, some viruses wipe out, there was one virus that wiped out 80% of anyone that was between the ages of 18 and 30 were dead people. And it's because their immune response was so strong that this virus selected against the strongest. So this is a classic kind of septic shock syndrome. They call it a cytokine storm. This is what takes so long to develop a vaccine, is they're really worried about the attenuated virus or any attenuated vaccine causing a cytokine storm in some individuals. You give them a vaccine and then they're dead. That's really why it takes so long, because you have to survey a lot of different people. Since we don't have clones to be able to test this on, and since we're not clones of each other, this is what caused a lot of time. Okay, back to the exotoxins. These are toxic proteins released from the pathogen and affect distant sites. Okay, and then one more, enterotoxins. These are exotoxin that affect small intestines. It's very safe to always associate intestines. Think of enterotoxins, you want to think of diarrhea. And promote fluid into intestines. Intestinal lumen, I don't have time, I don't have the right. So this is going to read small intestines and promotes fluid into intestinal lumen. Vomiting and diarrhea would be your symptoms. I don't know why I love the next several things that we're about to talk about. It's one of my favorite things to talk about all semester. Okay, so you guys got that. All right, we're going to go back into this. Pay attention now. A lot of mechanisms coming up, very pertinent for this exam. You can almost say that we've been waiting to get to this point. Okay, so we have AB exotoxins. A, attack, B, bind. We're all experts on that. We're going to talk about different examples now. That is a bunch of different AB exotoxins. I think the only thing you should be able to take away from this is there's a lot of different types of exotoxins, okay? AB exotoxins. They also have cytolytic toxins and superantigen toxins. We'll talk about this. So I've introduced AB toxins. I'm going to briefly go over cytolytic toxins, and then I'm going to go over cytotoxins. So I'm a little bit backwards. Okay. So we have diphtheria exotoxin. So that's something that you should put in your notes as a topic to cover. That's figure 2512, diphtheria exotoxin. So this is an AB cytotoxin. And this blocks protein synthesis. So follow me here. This is something I couldn't do as good on the board. So we have AB, right? So what does B do? B binds to the receptor. What does A do? Goes into the cell. And what A does is it basically it adenylates the elongation factor 2 subunit, or protein, I should say. So here you have an ADP event. So sorry, I thought I saw, oh yeah. So this is if this is confusing for anyone, ADP ribosyl group, just ADP, it's fine. That's what's happening. Let me remind you. So this is this hitchhiking structure. This is a ribosome. Ribosomes translate proteins. You remember that. Elongation factor 2. So if you remember, the A site receives transfer RNA, right? The transfer RNA is escorted to the A site by elongation factor 2, right? So elongation factor 2 is an essential protein that functions to escort transfer RNAs into the A site during translation. What the A subunit of diphtheria exotoxin does is it adds an ADP to elongation factor 2. This inactivates elongation factor 2, and now you can no longer escort transfer RNA into the A site during translation. Okay. Say it again, because I know some people are like, well, can you say that again? I can hear you. can hear you saying that to me. So we have diphtheria exotoxin, an AB exotoxin. So we have two subunits, and we can predict what those functions will be. The B binds to the receptor, A causes the disease, attacks, right? So the A comes into the cell, and it binds to elongation factor 2. Elongation factor 2 is a protein that functions to escort transfer RNA into the A site during translation. The A subunit of diphtheria toxin adds an ADP to EF2, and that inactivates elongation factor 2, EF2. In this inactivated state, EF2 cannot escort transfer RNA into the A site during translation. So you block translation. That's what happens. That's how this thing hurts you. Any questions on this? Now we're going to talk about neurological exotoxins. Clostridium, which is a spore-forming gram-negative, and it affects nervous tissue. There's two different mechanisms. Hold on a minute. I'm making this for you, so I'm going to make it nice for you. Live editing, Dr. Cruz, okay. I did all this yesterday. I'm so frustrated that I all got erased. Anyway, so what you have is a normal, here on the left, what you have is a normal neurological, neuromuscular event where you have a motor neuron. Motor neurons stimulate muscle cells to contract. And normally what you have is that you have the neuron releases acetylcholine. Acetylcholine binds to the muscle cell and a contraction occurs. This is normal. Neuron, acetylcholine, binding, muscle contraction. What happens with botulism is that botulism blocks the release of acetylcholine from the motor neuron. And without acetylcholine, the muscle does not contract. This is the basis for Botox. This is just localized botulinum toxin, so that you can look expressionless for life. I think that's why this is just my theory and conjecture. Everyone's talking about how Joe Biden, our presidential candidate for the Democratic Party, how he seems like he's lost it or he doesn't know. I think he's fine. There's nothing to indicate that he's neurologically has any issues. I think what it is he's taking Botox. I think that's totally what it is. So he looks expressionless. This is really common for people that are older and suddenly expressionless. I don't think they look younger. I think that if you look, have wrinkles and are vibrant, but ladies are rolling their eyes. They're like, yeah, right. But this is really this has a lot of therapeutic benefits. There's a lot of instances on diseases being able to go in a different direction due to this treatment, right? Overexcited areas of the body that are due to spasms or muscle contractions. All right. So this is the mechanism of botulism. The toxin. This is a toxin that blocks acetylcholine release from motor cells, and then the receptor. There's nothing. The receptors on the muscle cells don't receive acetylcholine, and there's no contraction. Oh. Sorry, I just can't stand the non-title slides. Okay. So here we have, just pay attention to the normal situation on the left. Oh, wait. Okay, let me unmute myself. I just wanted to go over, this is a part where I go over some muscle physiology. Okay, so you guys see my arm, my nice arm? Okay, so yeah, this is my bicep, right? Let me bring it over here. So this is my bicep. So this is how muscles work. So if I contract my bicep, my tricep has to be relaxed. There's coordinated neuromuscular signals that have to occur in order for me to just simply like raise my arm up. If I want to go down, if I go down with my arm, then I'm contracting my tricep, and there has to be concerted neural signal to relax my bicep. If I keep my bicep contracted to its potential while this is going, you'll have a muscle tear. So your muscles are constantly coordinating in the sense. So that means that when I contract in this direction, there has to be a relaxation. So uh, the mechanism to induce this relaxation comes from the activity of accessory neurons called inhibitory interneurons. So this is the normal motor neuron, right? And then what you have is you have this inhibitory interneuron. And so when it's time for this muscle to relax, so I'm flexing my bicep, now the tricep has to relax, the inhibitory interneuron will release glycine that binds to the motor neuron. This glycine binding to the motor neuron tells the motor neuron stop producing acetylcholine, and this relaxes the muscle. Okay, so I'll go again. So if I need this muscle to relax down here, I need this motor neuron to stop producing acetylcholine. So that happens when this inhibitory interneuron releases glycine. Glycine hits the receptors on the motor neuron stops releasing acetylcholine, then we have muscle relaxation. That's a normal event. Tetanus exploits this by releasing a toxin that it actually, so if you have a tetanus puncture wound anywhere on your body, what will happen is this toxin will travel, it's called retrograde migration, travel up your neurons. It's so crazy until it gets to your spine. And then from there, what it does is it inhibits the inhibitory interneurons that tell the motor neurons throughout the body to, so normally, if everything's functioning okay by the spine, you are relaxing where you're supposed to be relaxing, flexing where you're supposed to be flexing. When you have this tetanus toxin, it stops glycine production. And so there's no signal on any motor neuron to stop contraction or to stop acetylcholine production. Since these motor neurons throughout the body are never told to stop producing acetylcholine, your body goes into a state of complete contraction. If left untreated, this leads to cardiac arrest, or it can lead to not a cardiac arrest. I'm having a blank when you can't breathe anymore. Um, Respiratory arrest, I think. I don't know if that's the right term, but basically your diaphragm becomes completely, it just stays in the contracted state and you're done. And it will go to your heart too, but it usually affects the diaphragm first. So that's why tetanus shots are so important. Anytime you have a puncture wound, anything that goes deep, especially metals, tetanus likes to live in soil. Clostridium species are commonly found in soil. And so if you have a dirty puncture wound, you got to go get your tetanus shot. Clostridium tetani is all over the soil. And I know of a person that succumbed to tetanus. You can get treated though. It's you don't want to be in the hospital. It probably doesn't feel too good. Okay. Any questions on these topics? So these are very important to me. Okay, the mechanisms of botulism and tetanus. All right, let's talk about enterotoxins. Enterotoxins, as we put before on our definitions, are these are exotoxins that affect the small intestines. All right, and this usually refers, you can just think diarrhea. Enterotoxins, diarrhea. It's synonymous. Okay. We'll talk about enterotoxin, cholera enterotoxin. This is a toxin that was going on during the Civil War. Could you imagine? Wars are really a lot of times when pandemics happen because you're combining, you're creating susceptible hosts, and you're have a high concentration of people, and you're also usually traversing, you're mixing populations. So during the Civil War, we had a lot of cholera. During World War One, we had the 1918 flu pandemic. As long as we can stay out of war, we should get through this COVID thing. All right, so let's go through the mechanism here. Oh God. Okay. I don't know how much physiology you guys have learned, but across the intestinal epithelial cells, um, your body is controlling salt balance on both ways, in both directions, on both sides of that membrane. This enables the cell to deliver fluids or restrict fluids into the intestinal lumen as needed. It's a function to draw nutrients into and across the epithelial small intestines because we take in nutrients that way. I don't want to get too much into physiology, but this is just a central mechanism. So what happens is normally ion movements can occur across the epithelial cell. In the case of the cholera toxin being present, Vibrio cholerae, here we have AB, enterotoxin. Enterotoxins are a type of exotoxin. It's just if it's in the small intestines, it's also known as an enterotoxin. AB subunit, B subunit. So here we have a picture of the Vibrio cholera cell, binds, releases the enterotoxin. The beta subunit must be depicted in green. Yeah, it is. So B binds, A attacks, right? So the B subunit binds to this GM1 cell surface. It's just a place to bind and dock. It doesn't really have physiological consequence except for the fact that it's a binding event. Now, this AB toxin has access to the cell to mobilize. And so these A subunits go to work. These A subunits activate adenylate cyclase. If you remember before, we were talking about, what was it? E. coli, lactoperon, adenylate cyclase makes cyclic AMP. Anyway, so the A subunit activates adenylate cyclase. That makes more cyclic AMP. So the one is the consequence of having a lot of cyclic AMP inside of an epithelial cell in the small intestines. Well, this tells transporters to go ahead and allow chloride and the CO3 bicarbonate anion across the lumen, across the epithelial cell into the intestinal lumen. So if you get all these anions going into the intestinal lumen, water will follow. So I'll just go through this again. We have cholera, AB, enterotoxin. Beta binds to the GM1 receptor. Now we've immobilized the AB enterotoxin. A attacks, it goes in. The A subunit stimulates adenylate cyclase activity, producing cyclic AMP. Cyclic AMP signals epithelial cells to put chloride and bicarbonate anion into the intestinal lumen. Water follows high salt concentrations, and so then you get a lot of water into the intestines. This leads to severe dehydration because you lose all of your water through diarrhea. In the 1800s, they would have cholera. They would have seasons where you had to boil your water. They would have like cholera season um, that um, would just be in your tap. You couldn't drink your tap water. I think this is one of the ways Tchaikovsky, the composer, died. He just willingly, I don't, it's not very dramatic. Maybe you can put his dramatic music to the background of thinking about this image, but then he just so dramatically grabbed a cup and went it to the faucet and filled the cup with water and downed it, and then he died. But not before a lot of diarrhea. Anyway, um, okay, so let me see how much time we have. I think we have enough time. We're a little over, but we have a couple concepts here that are really good. So we have cytolytic exotoxins. So these kill cells by attacking the membrane. Okay, work by degrading cytoplasmic membrane integrity, causes cell lysis and death. Hemolysis is when you do red blood cells. If you kill nucleated cells, then Staphylococcus alpha toxin kills. So anyway, here's blood agar. If you have strep throat, what you'll be doing is that you first look under a microscope and you look for gram-positive cells, and then you look and then you plate the cells on a blood agar plate and see if you see lysis. This beta hemolysis activity is very characteristic of positive strep throat test. So how does it work? Here what we have is the toxin pore, and this is this toxin is secreted by the bacterial cells. And how it works is this subunit will, so if you picture just one subunit entering the cell membrane, boom. Then another subunit will go on, but they have an affinity for each other. And so what happens is it binds to there, but they don't align linearly. They have a slight curvature to them. If you remember endocytosis, there's some remnants of those mechanisms present in this diagram. And then as you add subunits, they will continue to form this semicircular pattern until they're all in this structure. Once there, then there'll be a plug of membrane that's in the bottom, in the middle here, boom, pops out. And then what happens is now you've just popped a hole in the tire, and that's a pretty good analogy because the average internal pressure of a biological cell is about the same as a tire pressure. So the minute that you introduce this kind of hole into a cell, it just all of the cytoplasmic contents are just going to leave, and then you get lysis of the cell. Superantigens cause overstimulation of the immune system, lead to shock and death, sepsis, generally due to localized infection, but systematic effects. So this is just more about how your immune system responds to them. And so we talked about this endotoxin structure in biology. So the lipo-LPS portion of the cell envelope of gram-negative bacteria is the toxin that ends up killing you, the LPS layer. Remember that? This is generally less toxic than exotoxins, but still it's pretty bad. A lot of people suffer through toxic shock through those things if you allow a lot of cultivation of these bacteria in anywhere in your body. I don't really care about this too much. It's a little interesting, but I don't have time to cover it. So just gram-negative bacteria, they have this LPS layer. When presented into the immune system, causes shock through the immune system. Okay. I think that's it. So is there any questions? Any questions at all?
I think I'm just gonna rewatch the video again. I got some good notes, but I think there's a lot of good stuff in there.
Yeah, I wish that we had one more session, but this is all very interesting. Very interesting. And I usually have time to go in a little more detail on some of these things and bring in my own examples. I'm going to rally around the book this time, but there's nothing wrong. You guys aren't getting less. I mean, this is really good material. I had to kind of not cover some of the metabolic stuff, but I think that you guys will not only enjoy this stuff a little bit better, it'll be easier to remember. It's better to put an exam on these concepts. I think you guys will enjoy it. Obviously, this exam will be chapter 25 heavy. Next Tuesday, I will have a session that will be review on a lot of these things. And so we'll get a good broad overview of a lot of these concepts. So it's not a last chance to get these concepts. It's very interesting. It's very, very interesting. And you should read up on I have a buddy of mine that works for the company that produces botulism, and there's all kinds of applications to it. It's very interesting. Anybody have any questions? Or you're like, Jesus, done. So I would say this is, sad because we're not going to see each other anymore. We're not seeing each other anymore. But we're going to have 2 review sessions next week, so this isn't the end at all. And so I hope that a lot of you will show up to those sessions. But I will say that I am sorry that I wasn't able to be physically with you all semester. I didn't see a pandemic coming. Kind of had a suspicion in January. I didn't say it until February. I got into the country from China a week. I came in literally 1 week before they closed the borders. I would have had to get a private plane to come pick me up. China was not letting any airplanes out. So I was worried about this a little bit, but I didn't think it would get this fast, this quick, but here we are. So I miss seeing you guys. I'm sorry that I can't be with you guys. I love teaching all of you, and it's been my pleasure, for sure.
Dr. Cruz, I have just a couple questions.
Yes. Go for it.
Wait, what's my question?
So there's
Oh,
one.
yeah, I remember.
Hold on. There was one thing I didn't have time to do, and this is important. When you guys come in on Tuesday, I'm going to have one of my students present a short bit on a disease. It's about 10 minutes long. It's very interesting. I just didn't have time to squeeze it in today. But Unu, I didn't forget about you. But we're not completely done. Tuesday and Thursday we're going to have sessions, and so we'll do that next week.
Okay.
Okay, question.
Okay, so I was looking at this photosynthesis thing yesterday, and I could not find the electron acceptors for cyclic process. Is this just there is no electron acceptor since it just recycles and goes around and around? Is that why?
That's right.
Also, do we say the cyclic ones are anaerobic, or not necessarily?
Yeah, you could say that. That's right.
Okay. And what about the Calvin cycle? Do we say that anaerobic or not? Because we use the ATP and NADP
Oh, you
from
definitely.
the
It definitely
aerobic.
doesn't. It's not. Okay, when you say anaerobic, what do you mean?
Does not have oxygen as electron acceptor.
Yeah, the Calvin cycle has nothing to do with oxygen. What I was wondering if you implied is when I see the Calvin cycle, can I assume that we are in anaerobic conditions? No. Ox Oxygenic plants use the Calvin cycle, but more importantly, it's not just a phototrophic event. We have microbes, chemolithotrophs that use the Calvin cycle to fix carbon.
Okay. And also another question about this photosynthesis processes. So do you have to know that all the steps of all those photosynthesis, or we need to know like what is microbes' role in them? Which one is it more to study about?
I'm a little confused on your question when you said microbes.
Oh, I was just saying, like in biochem class, we have to know all steps and reactions and everything for this kind of mechanism. Yeah.
Every step the electron goes to, every carrier.
Yeah, and the reactions, like what becomes.
Oh God. The reactions are pretty good. What kind of reactions you guys go into? Plastic quinone cycling and stuff
Yeah,
like that?
those. Mm-hmm.
Really? Oh wow. Jeez. Man.
Biochem is a bear.
Oh my God.
So we don't need to know those things for this class,
No.
right?
No. What I care about is the fact that you have a photosystem, that you have antenna molecules that are directing light towards that photosystem, and that you excite this electron, and to know what that electron donor is, and then that this electron, as it falls in energy, is creating a proton gradient, and that ATP produced, and then NADPH produced is going to be used to convert CO2 to glucose. That's all I care about.
Okay, cool. Thank you.
Yes.
And so I just want to make sure we don't have to do packbacks anymore, right?
No, you're done.
Okay, thank you.
Yes.
Thank you, Dr. Cruz. Bye-bye.
All right, I'll see you later. Okay, unless there's more questions, I'm going to sign off here. All right, I'll see you guys Tuesday, and I'll try to get these PowerPoints up. I'm going to wait until the week of our final to put the quizzes out. So don't be shocked if you don't see quizzes this Tuesday, just because I think that you'll be studying for the final at that time. And I know you guys have other finals, and so I don't want to disrupt your study habits. So I'm just going to wait. Then Tuesday, we're going to have our first review session. That starts off with a presentation from one of our graduate students. Okay. All right, guys, you take care. Oh, yeah.
Do you still think it should be cumulative? Now we have so much information for it, just let alone the last part.
No. I thought I wrote notes on this before. No, I don't think it should be cumulative. Let me make sure that's a good statement. I get in trouble with these things. Yeah, we can do chapter. I can agree to that. Yeah, we can just do this as a exam 3. I can agree to that. I can agree to that.
Cool. So whatever we covered after.
Yeah, chapter 20.
Midterm.
Basically, we're going to be going from chapter 20 to chapter 23 to a little bit of chapter 14, where it covers. Yeah, chapter 14. Chapter 14, where I covered it, not the whole thing, and then chapter 25, what we just covered in the past couple sessions. Yeah, I'll just have a exam 3. I won't do a cumulative. It's too much material.
Hey, thanks.
Okay. All right, guys. See you Tuesday. Okay.
See you. Bye-bye.